All right. I want to pick up where we left off. Oops. For that I'm not connected here. There we go. All right, I want to pick up where we left off, right at the end of that profile of a cancer cell group of slides. We talked a little bit about apoptosis, but I want to do a little more detail on the process of apoptosis. Apoptosis is programmed cell death. The cell does this itself at an appropriate time. Some examples I gave you: the finger webbing during development of a fetus, to kill off the cells to give individual fingers. It happens at other points in development. It actually happens in neural tissue at one point in development where some cells are killed off, and it actually enhances connections between other cells and the development of neural tissue in a fetus. It's also used to get rid of defective cells. So virally infected cells will undergo apoptosis as a way to get rid of that viral infected cell. It also can be triggered because of DNA damage. So if there's significant DNA damage, that can trigger apoptosis. So this is an ability that all cells have that gets turned on in an appropriate time. So important in development and as a way to get rid of infected cells or cells with DNA damage. It is a highly regulated process. It's very different from the other cell death process that we see of necrosis. Necrosis is not programmed cell death. This is cell death from tissue damage, something along those lines, a wound. The big difference between necrosis and apoptosis is that necrosis usually eventually causes cell eruption and bursting. Why is that bad? That triggers an inflammatory response. So this tends to cause a bigger amount of damage, necrosis does, than apoptosis. Apoptosis is much more highly regulated. It happens on an individual scale. Necrosis is something you see because of tissue damage, massive infection, something along those lines is going to trigger necrosis, where the cell swells and bursts and causes an inflammatory response. Now in apoptosis, highly regulated. What happens here is when the cell gets the signal that it's time to go through apoptosis, first thing is the cell starts to shrink. It actually shrinks in size. It also starts to form what we call blebs. Blebs are these little kind of pinching off of pieces of the cell. It forms this blebbing activity. I think I opened those doors. Finally, what's happening is the actual internal cell structure is being dismantled. It's taking apart the cytoskeleton. It's taking apart individual organelles and actually pulling it apart piece by piece. Instead of just exploding it and letting the cell die, it's actually pulling it apart piece by piece from the inside out. It's also dismantling the nucleus in this process of apoptosis as well. It will degrade DNA. It will actually start to degrade its own DNA during this process. Why does it want to do that? Is it dangerous to release cellular DNA into the environment? Probably not. But what would be dangerous if there was other kind of DNA in there? What if it was virally infected and you release viral DNA accidentally, or you release intracellular bacterial DNA, things like chlamydia or bacteria that infect inside cells? So you could be releasing the DNA of a pathogen, and how they get around that is they degrade DNA in the cell before the cell finishes apoptosis to get rid of any of that foreign, possibly dangerous DNA. Eventually, these blebs that are formed, we call them apoptotic bodies, and those are absorbed by phagocytes in the area. So white blood cells that normally look for things to destroy, they're normally looking for bacteria and other infectious agents, they will also ingest and completely degrade these apoptotic bodies. So it's a very regulated process. Slowly dismantle the cell, get rid of any DNA that's inside of it, degrade that DNA, pinch off the blebs, and then have phagocytes get rid of the rest of that material to get rid of these cells. Now the process of apoptosis, again highly regulated, is run by a group of proteins called the caspases. So here on a molecular scale, what's happening in apoptosis? It's a signal cascade. I feel like there's a common theme here. Everything's a signal cascade. That's what's happening here as well. There is a signal cascade that relies on these proteins called the caspases. Caspase proteins, when they are inactive, they're already present in the cytoplasm. They're called procaspases. They get cleaved to activate them into this caspase form. So, inactive precursor bases in the cytoplasm. They generally are hanging out in their inactive form in the cytoplasm. When they get activated, they get cleaved into this active caspase form, and that triggers this cascade. One caspase activates the next caspase, which activates the next caspase. There's about 8 or 10 of them that go down a signal transduction pathway where they're activating the next one, the next one, and the next one, and eventually they result in triggering apoptosis. They do that through turning on gene expression, beginning the dismantling of the cytoskeleton, all of those activities in order to trigger the beginning of apoptosis. So cascade of caspase proteins, each starting as an inactive procaspase that becomes activated when it's converted into its caspase form. Now, how does the caspase cascade get, like a tongue twister, caspase cascade. How does the caspase cascade get started? Well, one way is actually through a ligand binding to a receptor, and they call it the death receptor. It's present there in the cell membrane. It's awaiting a signal indicating it's time for this cell to die. What kind of eventualities would release a ligand, meaning death? Infection. If there's an infection in nearby tissue, it's possible it will send out a death ligand in order to trigger killing of the cells in the area so you don't get spreading of that virus. That's one possible reason. What else would want a signal transduction cascade to trigger caspases?
Maybe it releases it itself.
If that cell releases it?
Yes.
Possibly. Sometimes if a cell is releasing something that another cell recognizes as being a cancerous protein or something like that, it can release a death ligand. There's some evidence to show that. Development, death ligands are released during development. That's how the finger webbing cells go through apoptosis. There's this release of that ligand that kills off all of the cells in that area. So this is normal. The death receptors function during development. They function during normal infection control, including, I would include in that, some cancer ability. They'll recognize cancer sometimes and cause that beginning of apoptosis. The other possibility, I've got that on the next slide, so let me put that here. So the external pathway here, responding to external signals, can trigger apoptosis through this caspase cascade, responding to signals from the environment. There's also an internal pathway that responds to DNA damage. Okay? Responds to DNA damage, and it will eventually stimulate the same caspase cascade and eventually stimulate the same issues and the same dismantling of the cell. Now, how this happens, how this happens is DNA damage will trigger a change in P53. So let me draw this out here. When there's significant DNA damage, significant DNA damage will cause a rise in a protein called P53. So the levels increase in in P53 levels. This protein, P53, is important because it activates another protein. It's not as vital in our talk about cancer, but that protein actually starts to make holes in the mitochondria. So, activates a protein that degrades the mitochondrial membranes. DNA damage triggers a rise in P53, which activates a protein that's going to degrade the membrane of the mitochondria. What kind of activity is happening in the mitochondria of a normal cell? What's happening? What does the mitochondria do? Making ATP. Remember the thing I think all grade schools memorize the same phrase. It's the powerhouse of the cell, right? I don't really know what that means, but all grade schoolers memorize that exact same phrase. It's the powerhouse of the cell making ATP. So you have electron transport chain activity, you have chemiosmosis happening at high levels inside of the mitochondria. That's really its specialized function. Part of the electron transport chain is this molecule called cytochrome C. Cytochrome C is leaching out of the mitochondria because of the membrane damage. Membrane damage to the mitochondria allows cytochrome C to be released. Cytochrome C activates the caspase cascade. So all roads lead to the caspase cascade and apoptosis. That's how apoptosis is triggered, whether it's through a death receptor from an external signal saying it's time for that cell to go, or it's internal recognizing DNA damage. P53 increases. Eventually you get degradation of the mitochondria. Cytochrome C is out in the cytoplasm, and cytochrome C triggers that same caspase cascade, leading to death and destruction of the cell through apoptosis. Question?
What is the cytochrome C is just in the mitochondria?
It's actually part of the electron transport chain. Remember the electrons moving through the different complexes. Cytochrome C is one of those complexes. So it's important in the electron transport chain, but in our purpose, what's really important is when it's in the cytoplasm, because normally it's not. It's actually membrane bound inside the inner membrane of the mitochondria. When it gets released, it winds up stimulating that same cascade. So it's a way of saying mitochondria is breaking down. We know that the mitochondria breaking down means there's likely to be DNA damage. Therefore, kill off this cell through apoptosis. So P53 levels rise with DNA damage, eventually leading to the activation of the caspase cascade through this cytochrome C molecule. All right. So if we do a little comparison here, again, back to the whole blue being normal cells, pink being cancer cells, normal cells will undergo apoptosis only if the correct signals are present, including cellular damage through the cytochrome C pathway in P53 here, DNA abnormalities, if they get the receptor signal from an outside source, etc. Cancer cells are abnormal by definition and do not undergo apoptosis, leading to proliferation of cancer cells. Cancer cells will not go through apoptosis, even when they have significant DNA damage. In some cases, even when they get the death ligand, they do not go through apoptosis. So they are resistant to this process. And we'll talk about a couple of ways they can do that next, being resistant to this whole process. We talked about P53 and its involvement in the apoptosis pathway. I want to talk a little bit more about how that is really used in a cancer cell. So again, normal in blue here, the P53 protein is there to detect altered DNA. It will arrest the cell cycle. In high levels, it will stop the cell cycle progression. It stops it so you cannot go through proliferation of cells with DNA damage. So p53 protein detects altered DNA, and it arrests the cell cycle, and of course it triggers apoptosis through the pathway that we just talked about. And the idea is to prevent the proliferation of cells that have DNA damage, right? You want to get rid of them. You don't want to proliferate these cells. In cancer cells, p53 is somehow damaged or altered. So if p53 is either not there, it's damaged, it's somehow altered, then the DNA damaged cells continue to proliferate, leading eventually to cancer. So many human cancers actually come down to some problem with p53. As many as half of all human cancers come down to p53 mutation. And that's across the board different kinds of cancers. So it's extremely common. This is the place where many mutations form that eventually lead to cancer. For one reason, because p53 is involved in that cell cycle. Remember, the cell cycle controls how fast they can go through cell division and proliferation. So if p53 is not slowing that down, they can continue to proliferate as quickly as they want to. And also, the DNA damage is not being monitored. So DNA damage can happen and the cell will continue to replicate. All of those internal controls blocking that process aren't there. Let's look at an example of how p53 can be altered and result in cancer. So here, this is sort of simplistic version of what we saw a moment ago. DNA damage triggers p53, through mitochondrial involvement. Eventually that triggers apoptosis in the caspase cascade. Well, here's a situation. So the p53 tumor suppressor gene is the most frequently mutated gene in all of human cancers. Half of all people diagnosed with cancer have a mutation in their p53 gene of some sort. We know that there are several things we've identified now. I shouldn't say we. I had nothing to do with this research. They have now identified carcinogens in tobacco smoke cause several p53 mutations in lung cancers. They've identified that chemicals in the smoke, the tobacco smoke, actually can trigger these mutations in p53. We know that sunlight causes p53 mutations in skin cancer. We can identify that that's actually the gene where those mutations happen and then result in skin cancer. We know that HPV, that's the human papillomavirus that causes cervical cancer. HPV causes p53 degradation in cervical cancers. Here's how it works with this particular infection. So what happens is HPV is a virus. It causes warts, all kinds of warts. All warts are caused by HPV. There are lots of different strains of HPV. They cause all different kinds of warts, including the general seed warts that show up on fingers and stuff like that. They cause plantar warts that happen in the sole of your foot. They also cause genital warts, an STD. Different strains of them form different kinds of warts. That's why sometimes they're one large individual one, and sometimes they look like cauliflower because there's so many teeny ones. If you've never seen pictures, just Google genital warts. You'll get plenty.
But can't you have the virus without having warts as well?
Yes. Some of them, your immune system, it really depends on the person's immune system more than anything else is what we think is happening. Although there are multiple strains. Some are more likely to cause warts, some are less likely to cause warts. There's 30-something strains they've identified now of HPV. So yeah, HPV is the cause for warts. Frogs have nothing to do with it, no matter what your grandma may have told you. It seems to be all caused by this particular group of viruses, the HPV viruses. It turns out that HPV can also trigger various types of cancer. Cancer of the cervix. Cervix, remember the opening to the uterus, that's the cervix. That touches the very bottom there is the cervix. Cancer of the cervix, as many as 95% of all cervical cancer cases are positive for HPV, which means this is the cause for cervical cancer. They've also seen it associated with throat cancers, penile cancers, and anal cancers. Those three kinds of cancers are far less common, so we don't have as good data, but it seems to increase the risk for all of those types of cancers as well. Here's how it works. It turns out that one of the proteins that HPV makes is called E6. E6 is a protein that will stimulate the destruction of P53. When it gets into those cervical cells, produces this protein E6, and it triggers the destruction of P53. So how does HPV cause cervical cancer? Its protein E6 degrades P53. And this is a song we see over and over again. They do it in different ways, but P53 is a common point to damage and then cause cancer, the inability to control your cell cycle or kill off cells that have DNA damage. So if HPV is making E6, E6 triggers the destruction of P53. Without any P53, what happens? No apoptosis, which means DNA damage goes uncontrolled. What else is no longer controlled? This cell cycle. That's the other thing that P53 is involved in. So you kind of have a double whammy. You have mutations that are never gotten rid of, right? But you also have replication of those DNA damaged cells because the cell cycle isn't being controlled. So it's kind of a double whammy where you get more likely to have mutations and you're more likely to propagate those mutations when P53 is no longer around. Right. We will actually come back to P53 later. We'll talk more about it when we talk about more tumor suppressor genes. Notice I say it's a tumor suppressor gene. There's a handful of genes that we found in the human genome that naturally suppress the formation of tumors. P53 is the big one here, and you know that he does that by getting rid of DNA damaged cells and controlling the cell cycle. He's the big one on that tumor suppressor side, but we'll talk more about tumor suppressor genes in a future lecture. All right, I want to switch gears and talk a little bit about how a cancer spreads. How cancers spread from the original tumor site to other sites. Factors that control this or must be there for this cancer to be able to spread. First is angiogenesis. Angiogenesis is the creation of new blood vessels. So increasing the vascularization. We'll talk a lot about that. But it turns out that any tumor that's going to grow beyond a few millimeters in size has to have a blood supply. So the cancer has to be able to trigger vascularization to itself in order to grow beyond a couple millimeters in size. And I'll show you the research that proved that. Some cancers invade local tissues. They stay localized into the area. They may spread beyond the original tumor site, but they're spreading through the tissues. We call that invasion. Other cancers will metastasize. Metastasize means that they're going to spread long distance. They do that by entering into the bloodstream or the lymph system so that they can move long distance inside of the body. That's metastasis. We'll see that blood flow patterns and organ-specific factors will determine where they set up their metastasis site. So where that tumor forms a new tumor will be controlled by blood flow pattern in the human, how our blood circulates through our body, as well as sometimes there's a particular attractant, and that cell will go to a specific tissue. And this is an overview. We're going to look at each one. The last one here, we probably are going to have to do in a separate topic, a separate lecture, is how the immune system influences cancer. Definitely there's been a lot of work on how the immune system interacts to slow the spread of some cancers, but in some cases there's a lot of work done on how the immune system interacts with cancerous cells and how they can detect it, how they can get rid of that, and sometimes how they fail. We'll handle that in another lecture. All right, so let's talk about angiogenesis compared to vasculogenesis. Two different terms that mean slightly different things. Vasculogenesis is when sort of a spontaneous creation of new blood vessels, really capillaries is how this starts. It's kind of a spontaneous event. What happens here is endothelial cells that are undifferentiated, they're just simply present in the tissue. These undifferentiated endothelial cells will assemble themselves into vessels. Now this happens most dramatically in embryonic development. where you have these precursor endothelial cells sort of just floating in the area. They will self-assemble into the vasculature system. We'll see there's a couple other places this happens as well, but mainly this is happening during early development. This is the formation of the original vasculature system for an embryo, is through this kind of random collection of cells, which is just amazing to think about how they can do that. More commonly, once we're beyond the embryonic stage, what we're talking about is angiogenesis. And angiogenesis is the growth and proliferation of endothelial cells from one vessel forming a new one. So here you have one vessel, you have the growth and division kind of pulling out of a new vessel. That's angiogenesis, where it's kind of branching off an existing one. That's more commonly what we're talking about once we get to, beyond embryo stage.
So, past embryonic development, we won't really have like undifferentiated cells.
Right. Right. We don't. No, we really don't have very many, at least not endothelial ones. There are some undifferentiated cells, and occasionally you see the crazy things like the cysts that form that have teeth and hair in them, right? Those were undifferentiated. So you've heard about that kind of stuff.
No.
Oh yeah, and people will have a cyst, and they go in, they get it removed, and it's like got a big mass of hair in it or teeth in it or something like that. And what that was during development, some of these cells didn't get into the right location, and so they're in the wrong location, and they start forming whatever it is they got signals to form. And then the inquirer says, you know, my secret twin was inside. Anyway, it's really fascinating. More interesting from the anthropomorphic stage. But anyway, so mostly what we're talking about is angiogenesis, which is extension from existing vessels to make new vessels and increase the vasculature in the area. Now, really, endothelial cells in an adult do not divide very commonly, because once you have the vasculature tissue that you need in a healthy adult without cancer going on, they really don't divide very quickly. In fact, the average endothelial cell divides once in 3 years. All right, so there's really not a lot of change. There's not a lot of increase in vascularization, except for very few exceptions to the rule, which I have on the next one. Angiogenesis is very, very active during development, of course, in the early process of development. Once you have the original blood vessels that were made through vasculogenesis, then you have kind of branching off, so very active during development. It can be stimulated when additional blood vessels are needed, so you can stimulate it during an injury, a wound response. You need new vascularization to the area. That can stimulate angiogenesis during wound healing. It is stimulated in menstruating women a couple days a month. You do have increased angiogenesis around the uterine lining. That's what causes the changes in the uterine lining with the menstrual cycle. And that's angiogenesis is stimulated a couple days a month in that particular location. That said, this is an extremely regulated process. There are both angiogenesis activators to trigger angiogenesis, and there are angiogenesis inhibitors that inhibit the process present in the tissues. So it's kind of a balancing act. In a healthy individual, the angiogenesis inhibitors predominate. Say that again. So in healthy tissue, the angiogenesis inhibitors predominate. So in order to get angiogenesis, the cancer cells are going to have to both make activators and get rid of inhibitors. So they got to do both jobs in order to get angiogenesis to occur. Because in normal, healthy tissue, these inhibitors are predominating, so cancer cells will have to get rid of them. All right, some of the early work here that showed the importance of angiogenesis. This was done by a guy named Judah Folkman. In 1971, he came up with this way to test his theory. It was identified that some tumors stayed very small in tissue and some tumors got very large. And they already knew at this point that the very large tumors had a whole bunch of vascularization around them, new blood vessels wrapping around them. So the idea was what came first, the tumor or the vascularization. And the predominating theory was big tumors formed where you already had blood vessels. That was the idea that was out there. his idea was different. He said maybe the cancer is actually triggering the production of the new blood vessels. So that was the question he was asking, is which comes first, the larger tumor or the blood vessels? And so here's what he did. He took this is a rabbit's thyroid gland. This isn't pretty research when we're talking about cancer, but this is the thyroid gland from a rabbit with its servicing blood supply here, hooked up in this container where nutrients are coming in one side. They injected cancerous cells into the thyroid. So what's happening here is the cells are in their preferred environment. They're getting nutrients, but they're unable to trigger new vascularization. Right? They don't have the ability to do that because of the limited blood supply that's here. And what he found is that when these tumors were grown in this isolated system, they grew to about 1 to 2 millimeters in size, and that's it. The tumor could not get any larger. But if you took the tumors out of this thyroid and put them into the rabbit's thyroid, in an actual rabbit, the tumors grew much larger than 2 millimeters in size. So there's definitely something happening inside of the rabbit that's not happening in this situation, in this controlled environment. So the next thing he did was he injected tumor cells into 2 different parts of the eye. I believe this was also rabbit. Yes. Into 2 different parts of the eye. He injected tumors into this anterior chamber. This anterior chamber does not have blood vessels servicing the area. All nutrients get to that chamber by simple diffusion. So there's no vascularization into that part of the eye. He injected tumors there, and he found that the tumors were no larger than 1 to 2 millimeters in size maximum. They wouldn't get any larger. But if he injected the exact same cells into the iris of the eye, which is serviced by blood vessels, the tumors grew enormously in size. Exact same cells, injected into 2 different locations. So the evidence showed that what's happening is the ability for the blood vessels to come in and service that tissue. Here's some of his data. Tumor cells that were in the anterior chamber where there's no natural vascularization stayed quite small in overall volume here, while those growing in the iris increased dramatically in volume because they were able to get that blood supply in. Showing that tumor growth depends on angiogenesis. It depends on this servicing of blood vessels to the area. So tumors release signaling molecules that trigger angiogenesis in the surrounding host tissue. I'm going to show you some evidence for that one as well in just a moment. But tumors are releasing these molecules that stimulate angiogenesis in the tissue around them. Without these molecules, tumors cannot grow beyond a few millimeters in size. So they have to have the ability to trigger new blood vessel formation through angiogenesis. So here, a little schematic here, they're showing a small localized tumor. Right, that small localized tumor will grow no larger than the evidence shows 2 millimeters maximum, will grow no larger unless it can trigger angiogenesis and get vascularization to the area to increase its nutrient feeding. So it can't get any larger without that triggering. Now, how are they doing it? Here's another really pleasant experiment. On how they figured out that they're actually releasing some molecules that stimulate angiogenesis. This is a chamber. Chamber has a porous membrane surrounding it. Cells are inside, and the cells cannot get through that porous membrane. So what they did is they have the chamber, right, with the porous membrane around it. They put cancerous cells inside of that chamber. Cells cannot get through the pores. And then they put it underneath the skin of the animal. They put it actually into the animal itself. Implant it into, I don't have the location of where they did it, but they implanted it into the animal. And what they found is there, after a couple of weeks, there was increased vascularization around their chamber, indicating that the cells are releasing something small enough to go through those pores, which is triggering growth of blood vessels up and around the chamber. They did this with normal cells and did not see that increase in vascularization around it. So indicating that they're releasing individual molecules that are capable of stimulating this growth of new blood vessels. All right, so how do they do that? Triggering angiogenesis. You guys could probably predict at this point it's going to be a signal transduction cascade. It turns out that they're releasing some kind of a growth factor that stimulates angiogenesis. The most common one that we see is called VEGF, vascular endothelial growth factor, appropriately named because it's going to increase the growth of endothelial cells and increase the expansion of the vascular tissue. It is a very common growth factor to be released by cancerous cells. And this VEGF is going to trigger the increase in blood vessel formation and the angiogenesis into the area. So it works through a signal transduction cascade just like we've seen before. VEGF, released by the cancerous cells, binds to a receptor. It then triggers a signal transduction cascade that eventually changes the expression of genes in those cells. Turns out the cells, the change in the expression here triggers several things. We know cell proliferation is involved, but probably the most important one for angiogenesis is the production of these proteins called MMPs, matrix metalloproteinases. Why are these important? Because they break down the extracellular matrix. Remember, our cells aren't floating around in jelly. They're actually kind of cemented into their certain locations by the extracellular matrix, parts of the basal lamina, this kind of underlying tissue between organs, etc. There's a very dense protein structure holding all of the parts of our body and all of this together. They don't kind of swim around in there. So in order for new blood vessels to form, you have to break down some of that tissue so that that blood vessel can form. That's done through these MMPs. So it breaks down part of the extracellular matrix, which is going to allow angiogenesis to occur. So it's stimulating the production of these proteins, this VEGF, which will lead to cell proliferation, breakdown of the extracellular matrix, both of which are going to be involved in the formation of new blood vessels. You can't do that unless you've broken through that extracellular matrix and you have allowed the cell proliferation to happen in those endothelial cells. Does that make sense? So two things are happening there to trigger angiogenesis from it. Now, this is the activating signal. This is an angiogenesis stimulator. So this is the activating signal that comes from the cancerous cells, the angiogenesis stimulator. It turns out that angiogenesis is a complex event. The complexity of angiogenesis here, you have to have a growth factor, like the VEGF we just talked about, released by tumor cells to stimulate the local endothelial cells to form new vessels. That's half of the equation. For angiogenesis to occur, you have to have an activator, and you also have to get rid of an inhibitor, because remember there are inhibitors naturally in the tissues to prevent this from happening. So the tumor must also overcome normal angiogenesis inhibitors found in the tissues. And I want to remind you that normally in healthy tissue, the inhibitors predominate. They are at a very high level because they're avoiding this exact situation. So you have a balancing act from a cancer cell's perspective. I need to make the activator and I need to get rid of those inhibitors. Two different things need to happen for this process to occur. So the tumor must release angiogenesis growth factors, and it must also deactivate inhibitors in order to induce the growth of new blood vessels. Let's talk about the other side of the equation, those inhibitors now. Inhibitors of angiogenesis. Inhibitors of angiogenesis, you probably have heard of them before. The most common ones are angiostatin and endostatin. These are both inhibitors of angiogenesis found naturally in the tissues. We find them at high levels in healthy tissues. This is an interesting study that was looking at the effect of angiogenesis inhibitors on tumors. So what they did, what they did is they grew cancer cells until they were 10-day-old tumors. So they had a 10-day-old tumor. Once they had that 10-day-old tumor, they injected endostatin, an angiogenesis inhibitor. And a couple of, This is 40 days later, they injected another round of endostatin. And then what they found is after 2 injections of the angiogenesis inhibitor, that tumor never was able to grow in size again. So what I'm trying to get to is angiogenesis inhibitors prevent the tumors from growing beyond that 1 to 2 millimeters in size. Right? Remember, they cannot grow 1 to 2 millimeters in size unless they have good angiogenesis, unless they have a good blood supply feeding them those nutrients. So, tumors must make growth factors, right, to stimulate angiogenesis. And degrade those normal angiogenesis inhibitors that are found in the tissues. Genesis inhibitors to allow angiogenesis. And grow beyond about 2 millimeters. So they've got to do both. They've got to get rid of those inhibitors, and they have to make their own activators. So again, what's happening here is they're testing the effectiveness. Is it possible to limit a tumor's growth just with these inhibitors? So what they did is they had tumors. They grew this tumor to 10 days old. They then injected the tumor into the animal. And what they did is at day, it's around day 20 if I remember right, they injected their inhibitor, right, their angiogenesis inhibitor. Sorry, it's at day 0 because this is at days after treatment. So they injected their angiogenesis inhibitor. They let it go for a little while, and you see there is an increase in tumor volume. Right? When they first put that inhibitor in, there is an increase in tumor volume. Eventually it dies off. They inject another round of the endostatin, their inhibitor, and there is a little increase in volume, and then it drops. And it's no longer able to grow in size no matter how long you leave it. So 2 treatments with the angiogenesis inhibitor was enough to stop tumor growth. Is that exciting? Right. Why? Yeah, so the next question is, so can we do that in a human? Right? Can we inject angiogenesis inhibitors into a human and possibly stop tumor growth? The good news to some extent is yes. There's several treatments that rely on this exact idea where they they use these angiogenesis inhibitors, or they try to increase your natural angiogenesis inhibitor levels. There's some evidence to show it works in some kinds of cancers, not as well in other kinds of cancers. And here's an interesting caveat. It turns out, if one tumor gets really, really large, if he gets really, really large, he actually starts to release his own angiogenesis inhibitors, which seems counterproductive. So if one tumor gets large enough, if a tumor gets large enough, it will start to release both some angiogenesis inhibitors as well as activators. Some inhibitors with the activators, which I know seems counterproductive, but why would he do that? So he doesn't start? Well, why would an inhibitor? He would definitely want to release an activator, but why an inhibitor? isn't it? So you think he's going to eventually die on his own? I don't have a lot of evidence to back that up. What were you saying, Jenny?
Is it true that like other tumors also grow and like it's competition?
It's competition. He wants monopoly over that area. So eventually, that's what we think anyway is happening. So eventually, if he gets large enough, he'll start to release some inhibitors because he doesn't want any local vascularization going anywhere else. Right? He wants the major blood supply into that tumor. I'm anthropomorphizing that tumor, but you get the idea. The whole point is to increase vascularization to him, to his tumor, and try to block it from going to other locations. So eventually, if he gets large enough, he'll start to release some inhibitors along with his activators. Now, of course, from his perspective, he always wants to keep the activators at a higher level, but he will try to block any other vascularization from happening in a nearby tissue and try to stop that from happening. Let's look at some treatment studies that they did on mice at the beginning of this work. So on the left-hand side, what they're doing here is they're injecting, these are cancer cells, these are lung cancer cells, yes. Lung cancer cells injected into a normal mouse and then into a mouse that is angiogenesis deficient. It's not capable of undergoing angiogenesis, it's a mutant. Not surprising, you inject the lung cancer cells into a normal mouse, you wind up with lung cancer. You inject those lung cancer cells into a mouse that cannot undergo angiogenesis, and you wind up with no cancer, no lung cancer, because the tumors can't go beyond a couple millimeters in size. Sorry, just reading my note to myself. Interesting difference. Breast cancer results, lung cancer cells injected. Oh, I'm sorry. These were breast cancer cells. Breast cancer cells, this is what happened. The interesting thing I have here is when they did the exact same thing with lung cancer cells, what they found is you did get lung cancer, but the lung cancer didn't spread with angiogenesis inhibitors. So it was a little bit different, which is why they did the next experiment. So it worked with breast cancer cells, but with lung cancer cells, you did see tumor formation, but you didn't see spreading or metastasizing of that tumor. So here's the next experiment that they did. They injected the lung cancer cells into two different mice. Here into this mouse, you got the tumor forming. You then removed that primary tumor and injected them with angiostatin, that's an inhibitor, right, angiogenesis inhibitor, and you got few to no metastases. Very few tumors came back from that. But if they removed the primary tumor but did not treat them with angiogenesis, you got lots of different metastatic sites. So what it's showing us is not only does this seem to be involved with limiting the original tumor, if you have enough inhibitors, it also seems to reduce spreading and reduce metastasizing. And that's where it becomes really useful for us in human treatment. So inhibitors, and I mean angiogenesis inhibitors, right, reduce metastasis, these new tumors from forming. Question?
So in the second, with lung cancer,
Mm-hmm.
I know you mentioned breast cancer and lung cancer, but that was for the initial study.
Right, they initially did this with breast cancer, and this was their result. They did it with lung cancer, and they didn't find it to be as clear, which is why they did this additional work.
Okay, and so that's not with an angiodeficient mouse.
Right, this is with a normal mouse, yeah. So what they're showing with lung cancer, and probably this is true of other types of cancer, is if you remove, now it all depends on removing the primary tumor. If you don't remove the primary tumor, this is a totally different case. But if you remove that primary tumor and then treat with angiogenesis inhibitors, the likelihood of metastasis goes down dramatically. And this was the exciting news for possibly treating patients. The numbers here, the average number of metastases on a mouse that was not treated with inhibitors was 50. The average number of metastasis on a mouse that was treated with inhibitors was 2. So it is a dramatic difference in reducing these new tumors from forming. So this is really the genesis of maybe the useful way to use angiogenesis inhibitors is after we remove a primary tumor from a human, then we treat with them to reduce the likelihood of spread. And that's how most treatments are used today that use angiogenesis inhibitors, is to reduce metastasis and spreading of those tumors. Now, an interesting thing also came out of this idea, and this is the idea of tumor dormancy. Perhaps high levels of angiostatin released by tumors can cause tumor dormancy. I mentioned this when we talked about the fact that if a tumor gets large enough, it will release some inhibitors with activators, right, at the same time in order to control the blood vessels coming directly to it. It turns out that high levels of angiostatin released by tumors, and that's the situation here, right, when those inhibitors are being released by our tumors, perhaps causes tumor dormancy. And what tumor dormancy means is it inhibits the growth of those new cancers. So if you have one large cancer site, it will actually inhibit the growth of new cancers because those inhibitors are present in the tissue. Some inhibitors with activators and promote dormancy of new tumors. Sort of an interesting idea. If you think about it, what would happen if you just removed that primary tumor and didn't change anything in the chemistry of the body? You're actually going to wind up with more metastases than you would have originally because you removed the primary tumor. The primary tumor was releasing some inhibitors. So he was actually keeping some of these new tumors from forming. So if you remove that primary tumor and you don't treat at all, you actually could wind up with a worst case scenario. You removed what was controlling those new tumors. You guys with me? Okay. Because that original tumor is releasing these inhibitors, okay, he's actually creating dormancy in the new tumors. They can't form because they're being inhibited by his large levels of inhibitor release. But if you left him, mouse would die of lung cancer. So it's not a good option either, but you wouldn't get those new tumors forming. So the best option here is to remove the original tumor and then treat with the inhibitor. So you're keeping that inhibitor level high to try and reduce the new tumors from forming. All right. So let's talk about how they spread from one location to another. Invasion versus metastasis. Some tumor cells will directly migrate into neighboring tissues. We call that invasion. So they're in one tissue, they're moving slowly into neighboring tissues. That's invasion. Other cancer cells will actually enter into the bloodstream or the lymph and travel to distant sites to set up new tumors. That's called metastasis, where they're going to a new site long distance away in order to set up a new tumor. We're going to walk through this picture in several steps because it requires several different possibilities. Remember, normal cells don't want to detach from other cells, right? They want to be attached to somebody else, and they don't want to be too dense. So it's normal cell activity. So there's several things that have to happen for a cancer cell to go through metastasis. First of all, he's going to have to release from the tumor. He's going to have to get through the extracellular matrix, through the basal lamina to even encounter a blood vessel. Remember, this is all dense protein. All that structure that's in between organs, etc., is a dense protein structure. The basal lamina and then the extracellular matrix are on the outside of tissues. All of that is this dense protein. You don't just float around in there. For a cell to move, it's going to have to degrade all of that tissue before it even gets to a blood vessel. Then it enters into the blood vessel, travels wherever it's going to go, and then of course it's got to come out of that blood vessel, degrade all of the tissue to form a new tumor as well. Lots of different abilities to metastasize. It's not just simply they happen to wind up in the bloodstream. They've got to be able to get to the bloodstream, which is a huge ability. So let's go through the steps of how a tumor cell is able to do that. So it requires 3 steps. The cancer cell must invade the tissues and penetrate the bloodstream, which sounds easy but is not, because because it has to get through that thick protein. Oh, no, I don't want to back up right now. Thank you for offering. All right. So cancer cells need to get through that dense protein structure in order to get to the bloodstream, penetrate into the bloodstream. Then once they're there, they're transported by the circulatory system, different parts of the body. We'll talk about how it determines where it's going, but it'll wind up somewhere. Could wind up in the lymph, but we'll see that all roads lead to the circulatory system, even if it's in the lymph. And then, of course, it's going to have to leave the blood and then enter into the new organ site to set up a new tumor, where they establish this new metastatic tumor in the new location. So let's start out with step 1 here. Leaving the original tumor location and entering into the bloodstream. So cancer cells invade the tissues and penetrate into the bloodstream, this first part of this picture. There's a couple of things that have to happen here, one of which we already talked about. What was the protein that attached normal cells to a solid surface, and cancer cells don't have that protein? It's not on this slide. We talked about it last time. Yeah, it was last time. This is a protein that holds the cell onto the extracellular matrix so that it doesn't, this is why normal cells won't float around and rot. They have to attach to a solid surface. Integrin. Okay? Normal cells have integrin that attaches them to a solid surface. Cancer cells, by their very nature, don't have integrin. So lack of integrin. I'm going to say integrins because there's multiple in that group. But lack of integrins, therefore, they're not feeling compelled to be attached to this extracellular matrix. So not attached to the extracellular matrix. That's one thing that's involved in this process. Because remember, if this cell has integrin, all of the other abilities don't matter. It's not going anywhere. It's going to stay attached to that solid surface, the extracellular matrix. That doesn't have an integrin, then it's able to go through the next couple of questions. The next one is, how do cells hold together? Right? So not only are they attached to a solid surface, the extracellular matrix or the bottom of a flask, they also hold on to one another, and they do that through another protein called E-cadherin. This holds cells together. Lack of E-cadherin so not attached to other cells. This allows us past the first hurdle, which is, how do I separate myself from this tumor to even think about moving somewhere else? First off, how do I separate from the extracellular matrix? You can't have integrin. They have to have gotten rid of integrin for this to work. How do I release myself from the other cells in the tumor? They can't have E-cadherin. These are two proteins that are on the outside of the cell that must be gone for a cell to go through metastasis. So they must have gotten rid of them, mutated them somehow for this process to start. So cancer cells lack both integrin and E-cadherin. Therefore, they're no longer held together on the extracellular matrix. They're no longer held together in this blob of cells, this tumor of cells. They're able to separate more easily than any normal cell would. A normal cell can't do that because it's attached to one another through E-cadherin and to a solid surface with the integrins. Turns out some cancer cells exhibit chemotaxis. Some of you guys talked about chemotaxis in your posters. We didn't talk about it formally in class, but who knows what chemotaxis is? Movement according to chemical gradient, chemical concentrations. Taxis is movement in Latin. That's why they call it taxicab, by the way. So movement according to a chemical gradient. Those of you who used it in your posters, you were talking about the ability for a bacteria to sense a nutrient and swim towards that nutrient, or to sense a repellent and swim away from that repellent. Turns out other cells do chemotaxis as well. White blood cells in our bodies do chemotaxis. They can sense an invader and they can move towards the invader to destroy it. Turns out some cancer cells show chemotaxis ability as well. They have developed the ability to know where that bloodstream is and move in the correct orientation, which is fascinating. They don't randomly move through the tissue. They chemotax towards the bloodstream. So they're recognizing signals coming from the bloodstream and they're actually moving in the right direction, not randomly through tissues. It's a very directed process. The other question is, so how are they released? We know. How are they able to find their way? They're chemotaxing. And then how are they moving through all of this dense protein structure? They have to create proteases to degrade barriers like the extracellular matrix, the basal lamina. They need to be able to degrade all of that, and they do that by releasing proteases. They eat their way through the tissue, or degrade, I suppose is a better term. They degrade their way through those tissues. Proteases degrade the barriers. Like the basal lamina, like the extracellular matrix, they'll actually degrade the outside of the blood vessel so that they can move into the blood vessel. They'll actually degrade a little bit of those endothelial tissue so it can squeeze into the blood vessel. That gets repaired quickly by the blood vessel itself. So there's lots of abilities here. I'm always struck by how many things have to happen for a cancer to go through this process. If you think about all of these different mutations that have to happen, all of these different abilities that it has to gain in order to successfully go through this process. I mean, just simply movement here, it had to do so many different things. But even before that, remember, it has to ignore cell cycle control, etc. So there's several mutations that have to happen for the cancer cells to be able to do this function. All right, let's talk a little bit more about the proteases there. The proteases that are involved in degrading the basal lamina and the extracellular matrix. How this happens is the protease here is plasminogen activator. This is what's released by most cancer cells are releasing this particular protease. This is the protease that they release, plasminogen activator. Turns out that plasminogen is present in high concentrations in most tissues of the human body. This is a precursor molecule. We've seen this idea before, like procaspases was a precursor to the active caspase. So here we have this precursor molecule, plasminogen, present in high concentrations in tissues and fluids. The protease, when it's present, activates it to plasmin. Plasmin does the degradation. It will degrade and allow movement through the tissue. It's taking advantage of what's already there, this plasminogen, in order to get it activated into plasmin to degrade that protein structure. It turns out that this is also involved in the MMPs. Remember the MMPs? Where were they useful? What were they doing? They were degrading extracellular matrix and allowing the formation of new blood vessels and angiogenesis. They allowed that new blood vessel formation. And they're doing the same thing. They degrade proteins. So it's stimulating, it's kind of crossing over with another pathway here. It's degrading those proteins, degrading that tissue so that it can move through the tissues very effectively. All right. So once they've moved through step one here, they've gone through all of these mutations to be able to get into the bloodstream. Once they're there in the bloodstream, the next thing is, where do they go from there? If the cancer cell winds up in the blood, great, it's circulating through the bloodstream. We'll look at how it decides where to stop in just a moment. Sometimes the cancer cells wind up in the lymphatics. They'll wind up in a lymph vessel instead. That's okay. Remember that lymph vessels lead to lymph nodes. Lymph nodes are serviced by the bloodstream. So it doesn't matter. If it gets into the lymph system, it will wind up in the bloodstream because there's significant crossover between the two. In fact, if it even avoids a lymph node, If you remember the lymph system, you guys learned about the lymph system? Right, it's like a drainage system, draining fluids out of the tissues, and eventually it pulls those fluids up and it dumps all of the fluid back into the circulatory system right above the heart. So even if it misses a lymph node, it doesn't matter. All lymph fluid winds up back in the circulatory system. So however it gets there, directly into the circulatory system or through the lymphatics, it's going to eventually wind up in the bloodstream. Now, it's being moved around through the circulatory system. It turns out that relatively few cancer cells actually survive the trip through the bloodstream. The original study that showed this, not actually this one, but the original study that showed this, they took radioactively labeled cancer cells. So they radioactively labeled cancer cells, and they ejected them into the bloodstream. 10 days later, they found that very few of those cells were still alive in the bloodstream. So it turns out very few cancer cells survive the bloodstream. Why that is, we don't entirely know. It appears that the bloodstream is inhospitable to most cancer cells. So most cells that actually make it there die before they go somewhere else to set up a new tumor. Very few of them actually can survive that trip. Something in the bloodstream is inhospitable to cancer cells. Probably this has links to immunology, because immunology is most likely involved here. Our immune system is probably killing a number of these cells while they're in the bloodstream. We don't know the details of how that works, but we can definitely see that very few of them survive the trip. So the question was, what's happening? Is it a matter of some cancers are better able to survive and some cancers are not? Or is this all sort of Darwinism? There's variation in the original tumor and some survive and some do not. So what they did is a little study here. They injected this mouse with cancer cells, melanoma cells, and they got very few metastases in the lungs. Very few cells survive the trip to the bloodstream. We know that happens, right? So very few cells survived. They got just a couple of tumors here. So what they did is they removed those tumor cells and they injected them into the next mouse. And they found that the next mouse, even though they injected it the same way through the bloodstream, they wound up with more metastases than they saw before. If they took those cells and injected them into the next mouse, you eventually got more and more and more metastases each time you did this. So what they're showing is that original tumor was a heterogeneous population. Some cells could form metastases and some cells could not. So what happened is then you went here and you selected them out. Those cells that made tumors, you selected out and you injected into the next one. So you're self-selecting for those cells that can form metastases. So it turns out that the original tumor cell was a heterogeneous group where some cells can survive and some cells cannot. And so apparently whichever cells can survive set up those new metastatic tumors. So it is just like we see with natural selection in other organisms. Cancer cells undergo natural selection through this metastasis process. Very few of them survive the trip through the bloodstream. But once they have, you've now selected for those that are better at forming new tumors than the original. So, original tumor appears to be a heterogeneous mix of cells. Where some can metastasize and some cannot. And the evidence for that is we can select for those that are better at metastasizing. So the original tumor has variation. Some can go through metastasis, some cannot. It seems to be random which cells wind up in the bloodstream. Some of them survive to form new tumors. Some of them do not survive to form new tumors. Another work that was done to prove this is they took one tumor, right? One tumor out of an animal. I think it was a mouse as well. They took one tumor out. They separated it out into individual cells. Some of those cells, they injected them into various different animals. Some of the other animals got tumors metastasized and some did not. So it shows that the original population was varied, where there are some that can go through that process and some cannot. Further work on this has shown that in lung cancer, where a lot of this work has been done, in human lung cancer, we can find a pattern of expression of 17 genes that predicts metastasis. So in human lung cancers, there's a pattern of expression of 17 particular genes. A pattern of expression of 17 genes that can predict your chances of metastasis. So they can take that original tumor and they can look for expression of these 17 genes. And if it follows a predictable pattern, your chances of metastasis are extremely high. Those 17 genes, some of which we know what they do, some of we don't, those 17 genes, that particular expression pattern is linked to metastasis, which is really interesting. So this is looked at in lung cancers as a predictor for how likely your lung cancer is to go through the metastatic process. Since then, they've looked at it in other forms of cancer, and it seems to hold true for metastasis of both prostate cancer and breast cancer as well. Originally seen in lung cancer, and now they can use it as a predictor in those cases as well. So what I want to get to is, it ain't easy to go through metastasis. Right? You have to survive to get into the bloodstream, and then you have to survive in the bloodstream. And it's only a handful of cells that apparently can do that. We know in human cancers, these 17 genes and the particular expression pattern is predictable, and that metastasis is likely. Question? So this is survival through the bloodstream. The next one is where are they going to wind up? So how they enter into a particular location. It's interesting. What controls where the metastases will develop, where these new tumors will form? Two things that can control this. First off, cancer cells are most likely to get lodged in capillaries. Capillaries are the very tiny little extensions of blood vessels. Those capillary beds are where an individual cell is most likely to get lodged into the capillary, not be able to move, and then exit to form a new tumor. So anywhere you have lots of capillary beds, dense capillary beds, you are more likely to have a metastasis because they're small and literally the tumor cells get stuck in the capillaries and then migrate into the tissues. So anywhere that you have large capillary beds is a possibility. So cancer cells are most likely to get lodged in capillaries because of the size and then migrate into surrounding tissues. But we can do one better than that. The blood flow pattern, not platter, the blood flow pattern through the human body is predictable. We know the order in which organs are serviced through the circulatory system. We can predict where the metastasis will show up based on the next organ that has dense capillary beds. So metastasis often follows this blood flow pattern. The next organ in the blood flow pattern that has dense capillary beds is the most likely place for metastasis to develop. It makes sense. Okay. So wherever it entered into the bloodstream, the next organ where you're going to find those dense capillary beds is the most likely location to see a new metastasis developing. And we can see that time and time again with various different kinds of cancers. In fact, 2/3 of the metastasis pattern can be predicted using blood flow pattern and capillary bed density. So we can predict them based on those two things. If you have an original tumor, so for example, if you have original tumor, stomach tumor, right, is the original site, it's most likely to metastasize in the liver because that's the next dense capillary bed location in the blood flow pattern. 2/3 of human cancers we can predict just using those two pieces of information. Next organ that has capillary beds on that circulatory pattern. The exceptions to this are few and far between, but there are a handful. Some cancers metastasize to a particular organ more or less frequently than you would expect based on capillary density and blood flow pattern. So about 1/3 of them don't follow our rule of thumb. 2/3 follow the predictable pattern, capillary bed density, blood flow pattern. About 1/3 of them don't follow that pattern. They show up in unusual places that we would not expect based on those two predictions. It appears that some cancer cells are particularly attracted by a certain tissue. So this cancer cell is headed for that tissue specifically. Almost always it's because of a growth factor made by those particular types of cells. The cancer is attracted to it and it's chemotaxing to that certain tissue. The classic example here is prostate cancer. If we looked at blood flow pattern, a minute ago, if we looked at blood flow pattern, the most likely place for prostate cancer to metastasize is to the lungs or the brain, as we would expect it to show up. Those are the two most likely locations based on the pattern and capillary bed density. But in fact, overwhelmingly, prostate cancer metastasizes to the bones, a location we would not expect. A lot of research has gone into what's happening there, and they did several interesting studies. They would take a prostate cancer tumor, grind it up to separate it into individual cells, and then they would put those cells with various different other kinds of cells. So they would take a prostate cell and a liver cell and inject it together into a patient, or they would an animal. They would take one of these cancer cells and a kidney cell and put them together. They would take it with a bone cell and put it together. And what they would find is that only if they injected the prostate cancer tumor cell with a bone cell was it able to go through and form a new tumor and metastasize. So something that the bone tissue is making is what's exciting to these prostate tumor cells. We now know that what's happening is they're releasing a growth factor. So we know that the prostate cancer cells are actually moving to the bones in response to this particular growth factor. They're actually chemotaxing. So there are some that don't follow the pattern, but most of the time they follow this blood flow pattern and this capillary density question. So prostate cancer is a weird exception to this rule. They've done a lot of work on this particular one, where prostate tumor cells chemotax. They actually are moving in response to a growth factor released by bone cells. And in fact, a prostate metastasis appears to only proliferate if it gets the growth factor it needs from those bone cells. So it's very specific in its particular pattern. All right. So if we put these steps back together here of metastasis, remember the first step is it has to release from the primary tumor. It has to approach and get into a blood vessel. It has to release its attachment to the extracellular matrix. Those proteins are called proteins that attach them to the extracellular matrix. Integrins. It's got to get rid of the integrins. It has to get rid of the proteins that attach them to one another. That is E-cadherin. It has to then know which direction to go. Has to recognize the location of the blood vessel and move in an appropriate way. That's chemotaxis, right? Ability to sense where it wants to go and move in that direction. And it has to work its way through all of this basal lamina tissue and extracellular matrix and get into the bloodstream through the release of proteases, right? Proteases to degrade all of that tissue and allow it to move through and enter into the bloodstream. Once it gets into the bloodstream, life is not good. Vast majority of cancer cells that get into the bloodstream do not survive the trip. Very few of them survive the trip. Okay? Exactly what's happening there, we think it's the immune system's involvement. We don't know the details, but it's likely to be the immune system's involvement. Once it's in the circulatory system, those few that have survived the trip then have to exit through the circulatory system. It's the same thing. They're going to degrade basal lamina through proteases and eventually set up a new tumor, a new metastatic tumor. And how do they know where to go? Blood flow pattern, capillary density, or if they're chemotaxing to a particular growth factor, then they'll go to a particular tissue as opposed to anywhere. Questions on metastasis? This brings us to the topic of tumor progression. Tumor progression is the idea that cells can acquire traits that confer a selective advantage. Let me back up and say this, kind of relating it to something we talked about before. The idea that that primary tumor is a heterogeneous population of cells, right? That original tumor, the cells are not all identical to one another. Remember, they're undergoing DNA damage. So the likelihood is they have different mutations than the neighboring cell. They're not all the same. Rounds of mutation will create more and more variety in their subpopulation. As they go, you're going to see more and more variety as it happens. Well, it turns out that in order for that tumor to go through and eventually form, you have to have several eventualities happen. So cells acquiring traits that confer a selective advantage. They have to have an increased growth rate, right? They have to be able to outgrow other cells in the area. The increased invasiveness, in other words, they can chemotax, they can make their proteases, they can invade into new tissues. The ability to survive in the bloodstream, that's somehow avoiding the immune system. Resistance to immune attack, the ability to grow in other organs. They're going to have to be able to survive in whatever new site they wind up in. Resistance to drugs, right? If the patient is under some kind of chemotherapy. The evasion of apoptosis, the ability to survive and have that cell not die out. All of these have to happen. All of these traits make a cancer cell more successful than other cells. Over time, natural selection is going to happen. Over time, that original population has lots of variety and only a few cells may fit this bill. But the longer the cancer goes on, the longer the tumor progresses, the more metastatic sites you have, you're going to wind up with more and more cells that have this perfect cancerous ability. They have all of these things. They've gotten rid of all the integrins and cadherins and the ability to make proteases and the ability to survive the bloodstream. They've gone through all of these changes. They've broken down P53. They know how to stop the cell cycle. Eventually, you wind up with less diversity over time. You have selected out those cells that cannot do everything needed to form a tumor. So what we're getting to is, over time, cells become more aggressive. Over time, the cancer becomes more aggressive because of the natural selection that's going on. You are getting rid of the cells that can't do everything. They are being outcompeted by the more aggressive cancerous cells. This is why you heard of the phenomenon where someone has melanoma, they remove the melanoma, and then 6 months later they have horribly invasive lung cancer. Right? The tumor progresses. The cells get better and better at doing what they're doing. They become less heterogeneous, more homogeneous. So over time and with each metastasis, the cells have reduced their diversity, and they're more and more able to produce new tumors aggressively than the original population was. And that's a function of natural selection. Same thing that happens in other populations of cells. Over time, they adapt, and the whole population will become less heterogeneous. So kind of to wrap up here, interactions here between the tumor and host that are involved in invasion and metastasis. The endothelial cell, of course, the endothelial cell is vital for a cancerous cell. It has to stimulate angiogenesis in order to feed itself, to grow beyond 1 millimeter or 2 in size. The endothelial cell has to be involved in that process. So angiogenesis stimulators to stimulate that process, and of course get rid of the inhibitors for that process to occur. If we talk about what's happening with other cells that may not be cancerous, but in the area, the cancer cell has to release itself from other neighboring cells. So of course, involvement there in E. cadherin. The other normal cells may be releasing some kind of growth factor or inhibitors that can either inhibit or assist in the cancerous process, like the bone cells. They're releasing a normal growth factor, but unfortunately that growth factor is a chemotaxis activator by prostate cancer cells. And then the extracellular matrix, the basal lamina, normally functions as a barrier to movement, but for a cancer cell, it's got to have gotten the ability to release from that through the release of integrins and also break it down through the release of proteases so it can move through in order to trigger metastasis. Immune cells, we're going to talk more about that in the future here, but the interaction between cancer and the immune system, this probably, almost undoubtedly, controls the reason why few cancers survive the bloodstream, because that's where the main immune function is. Where the immune cells will be attacking cancerous cells in the bloodstream, the few that are able to withstand that attack are likely to set up new metastatic tumors. Questions on spreading of cancer? All right, let me upload this while I'm thinking about it.